Hi, students, and welcome back to the Sound of Science. Today, we're going to be talking about the structure of DNA. Now, this is something that we've talked about before when we talked about macromolecules, but we're going to go a little bit more in depth into it, and we're going to talk about how DNA is copied, how it's replicated. So, to begin, the stuff of DNA. DNA is going to be made up of this 5-carbon sugar, which we call a pentose sugar. It's made up of phosphates, and it's made up of nitrogen bases. And this should be fairly familiar. We talked about this when we did macromolecules, right? Those bases, the nitrogenous bases, can either be cytosine, guanine, adenine, or thymine. And then in cases when we're talking about RNA, which is just another type of nucleic acid, it's not going to have thymine. Instead, it's going to have uracil. So we'll get there in just a few minutes. But for right now, you just need to know that this is the structure of a nucleotide. This is how DNA is built. Okay. So back in the 1900s, early 1900s, we didn't know how DNA paired together. And so there was a lot of different scientists that were working on that. And one of those is this guy right here named Chargaff. Erwin Chargaff created Chargaff's rule, Chargaff's ratio, and it's based on the amount of nitrogenous bases in each of the DNA strands. So essentially what he figured out is that the number of adenines is the same as the number of thymines. And the number of guanines is the same as the number of cytosines. So what does that mean? It just simply put means A pairs with T and C pairs with G. And we're going to see that every single time. And so Chargaff was able to figure this out. And this is actually going to lead us to a better understanding of how DNA functions. Okay, so just so you can kind of get the overview here, we have the nucleotide, which you can see there at the top, it's been circled. And in between each side of the DNA, where the C's and the G's pair and the A's and the T's pair, we're going to have hydrogen bonds. It's important that you know that. In fact, take a moment and make sure that you have drawn out or sketched out this model. This model is really going to help you understand DNA. And when you're asked to label it later, maybe on a test coming up in a couple weeks, you'll actually be able to label those things. As far as the sugar phosphate backbone goes, so that is that part of the nucleotide that's the pentose sugar and the phosphate. And so we always consider that the backbone, with the nitrogenous bases making up the rungs of the ladder. So when we look at it in a flat form, we call that the DNA ladder. When we look at it in its three-dimensional form, it's what we call a double helix, because it has the two sides and they look like they've been twisted, like a twisted ladder. That's the double helix. All right, so looking here at our model, our double helix model, what you're going to see are anti-parallel strands, basically meaning that the strands are running in opposite directions. Okay, obviously that doesn't mean that the strands are going anywhere, but if you take a look at it, there are a couple of numbers to pay attention to. One says 5 prime, which is just that little mark behind it, and the other says 3 prime. So one side of our DNA is going to be set up 5 prime to 3 prime. What the heck does that mean? Essentially, it just means that the phosphate is always attached at the 5 or the 5th carbon in the pentose sugar. And so when we are looking at the pentose sugar there at the very top left, where you see the 5 prime on the blue side, you actually see the phosphate is attached to that 5th carbon. That's our 5th carbon in our ring. And if you look across at the yellow side, you'll notice that at the top we have a 3 prime. Literally, the pentose sugar, the whole nucleotide, is flipped upside down. This is how DNA is going to be built, and it makes a difference when we talk about replication. But for right now, it's not a huge deal. Just know that it runs 5' to 3' on one side, 3' to 5' on the other. And then again, this points out that there's weak hydrogen bonds between the nitrogenous bases, between the nucleotides, that hold the two sides of the DNA together. When we talk about complementary strands of DNA, really what we're talking about is what it pairs with. So just like we saw before, on one side of the DNA, you have, and actually I can go back and show you. Here we go. I can show you right here how the cytosines pair with the guanines and the adenines pair with the thymines. This is what we're talking about when we're talking about complementary strands. You always have your prime strand, usually the 5' to 3' side, and the other side is going to be your complementary side. So that's what we're going to be taking a look at here. So I want you to take a look, and I've given you half of a DNA molecule. The code here is ACGTTAGC. So you guys can see that, right? So the question is, what is the complementary strand going to be? Well, to determine the complementary strand, we literally just need to figure out what each letter pairs with. So A is always going to pair with T. C will pair with G. G will pair with C. And T will pair with A. And then as we move through, we can fill in the rest of our complementary strand, just like that. So you can see that while the ACGTTAGC side is our primary, our complementary strand is going to be the TGCAATCG that pairs with it. So you may be asked to come up with the complementary strand. You might also be asked to come up with a complementary strand in RNA, and we'll get there, and I'll show that to you as well. Okay, now I want to talk about how we found the structure of DNA. And this is a very important story, so pay close attention to the people that I'm talking about. You definitely need to know the people and really what their contribution was. So to start off, just a little bit of history. Before the 1950s, we don't really know what DNA looks like or what shape it's in. There's tons of researchers that are trying to figure this out, but so far, really no luck. So these two individuals, James Watson and Francis Crick, one's British and one's American, are working together in the Cavendish labs. They're trying to discover the answer to what does DNA look like? What is the structure of DNA? And they're actually taking models that they're constructing out of paper. They're cutting out paper models and they're trying to glue them together. And they can't quite figure out how everything is going to fit together. They know that there's nitrogenous bases, they know there's phosphates, and they know there's pentose sugars, and they know that they fit together in some way. They just can't quite figure it out. Meanwhile, in another lab area at Cavendish Labs, Rosalind Franklin is using X-ray crystallography. She's also trying to determine the structure of DNA. Rosalind Franklin was not easy to get along with, apparently. Most of her colleagues were not super fond of her, but she was brilliant, absolutely brilliant, far more intelligent than most of her colleagues. And in particular, some of her male colleagues felt very threatened by her intelligence and the fact that she was like, I don't need anybody to be a partner to me. I can do this on my own. Well, initially when she's hired, she was hired to work in a lab and to run a lab along with Lawrence Wilkins. And the two of them did not get along. They didn't get along at all. Franklin thought she was the boss, and Wilkins thought he needed to be the boss because he was not going to take directions from a woman, particularly one that he disliked. So meanwhile, Rosalind Franklin is using X-ray crystallography. And essentially what she's doing is she is crystallizing DNA. She is shooting X-rays at it, and she's taking photographs of that using this X-ray technology. Well, Wilkins became very annoyed with her, and he actually steals one of her photographs, and this is photograph 51. And it depicts the double helix shape of DNA. He took it without her permission. He gave it to Watson and to Crick, and the end result is that they are able to write a paper to be published in Nature discussing the structure of DNA. This photograph tells them what DNA looked like. They kind of came out later and particularly Watson said some pretty horrible things about Rosalind Franklin. That she didn't understand her own research, that she would never have come to that conclusion, and therefore they had the right to that photograph, despite the fact that what they did was completely unethical. So Rosalind Franklin does not win the Nobel Prize. Watson, Crick, and Lawrence Wilkins do about 10 years later. But part of the reason that Franklin doesn't win the Nobel Prize is that she actually died as a result of working with X-rays. She develops ovarian cancer, and she dies before the Nobel Prize is awarded. But almost like a slap in the face, the same issue of the journal Nature, where Watson, Crick, and Wilkins published their article talking about the structure of DNA, Franklin actually had written an article as well discussing the structure of DNA. And her article was published, but her article was published in the back of the journal, whereas theirs was on the front page. So it's not until later years that anyone really gives Franklin the credit that she's due for this discovery. Okay, so after that sad tale, let's take a different look and let's talk about replication. And this is just literally how DNA makes a copy of itself. So this is going to happen during the S phase of the cell cycle. And you guys have just learned about the cell cycle, so that should still be fresh in your mind. Remember, we have G1, S, G2, and then we go into mitosis and eventually cytokinesis. So this is during that S phase of interphase. So the DNA, to be copied, needs to be unzipped. And literally exactly what you see here in this picture. There's a molecule called helicase. And the DNA helicase is going to break those little hydrogen bonds, and it's going to separate the molecules of DNA from each other. The DNA polymerase is then going to come along, and it's actually going to help add these free floating nitrogen bases to the original DNA strands. So the reason that it does that is it actually allows it to make a complete copy. Remember, the DNA will only bond with the nitrogenous bases that match it. So A's are going to pair with T's, and C's are going to pair with G's. And so by doing this, they're actually able to make a copy. They're able to replicate it using that helicase and the DNA polymerase. And if you remember what I told you, anything that ends in ACE, ASE, is an enzyme. So both of these are going to be enzymes. Because they're using the template of the original strand, ultimately you're going to end up with two strands of DNA that are identical. Just a quick note about the difference between prokaryotes and eukaryotes here. Prokaryotes, like bacteria, have a single circular DNA molecule, and it's actually going to start at one portion of the DNA and it's going to work its way around. Eukaryotes, we're actually going to have little bubbles of replication happening all over the DNA at one time. Otherwise, it would just take too long. This is very, very complicated. Okay, so it takes a long time for that to happen. Okay, now this is the part that can get very confusing, and you need to make sure that you understand this. You need to write down essentially this entire slide. Put this entire slide in your notes, the picture and the thing with the arrows, okay? So what we're starting off with is our strand of DNA inside the nucleus. And your DNA is really important. Nothing bad should happen to it. If it gets a mutation, then all your DNA will be messed up. So in order to protect it, your cell is going to leave it in the nucleus like it's the VIP section. And the DNA is instead going to have a copy made, but this copy is going to be made in something called mRNA. mRNA is messenger RNA, and RNA is just another form of nucleic acid. We'll talk in a minute about the differences between them, but essentially, this is going to be a single-stranded molecule. And because it's single-stranded, it actually can fit outside of the nucleus. It can leave the nucleus through pores in the nuclear membrane. The DNA is just too big. It can't. So the DNA is going to be transcribed through a process called transcription into something called mRNA. That mRNA is going to leave the nucleus and it's going to go attach itself to a ribosome. So the ribosome then is going to help the DNA or the mRNA be translated into a protein. So how is the DNA going to be translated? Well, these little things called tRNAs, which you see kind of coming in, they're the weird looking things right here. These are tRNAs, and each tRNA has an amino acid attached to it. Each tRNA pairs with a segment of mRNA. So the mRNA segments pair again. A pairs with T, C pairs with G, and it's the same idea, with the exception of in the mRNA and in the tRNA, we no longer have T, we only have U. We have uracil as replacing the T. So they're going to pair up based on complementary sequences. And as they pair, they actually leave behind an amino acid. So what you're seeing here is a long amino acid chain that's forming. Remember back to macromolecules and biochemistry. We talked about how proteins are made up of amino acids. This is what we were talking about. These are the amino acids that are going to make our protein. And that protein is ultimately going to be twisted up into a different shape, and then it's going to be added to other twisted up amino acid chains, and ultimately we're going to get a protein. The shape of that protein is very important. If the protein isn't the right shape, it doesn't do its job, and you have a mutation or a trait that is potentially harmful rather than helpful. So I've said it several times now, but I'm going to show you again. So that DNA is going to be transcribed into mRNA. It's a complementary copy. And so when I told you earlier when we talked about the complementary copy or the complementary strand of DNA that you might be asked to do this with RNA, and that's exactly what we're here. So where G still pairs with C and T still pairs with A is now going to pair with U because there is no T in RNA. So you'll notice if you look here, all of the places where there was or where there would be a T are now U's. And if you notice too, we've got 3 letter segments marked off. These we refer to as codons. Codons, each codon is going to code for a specific amino acid. So they always work in these triplet pairs, these codon. To translate means to figure out what each of those codons stands for. So, and I'm going to go through this with you in class as well because I know it can be somewhat confusing. But if you take a look here, these are potential codons, and it gives you an abbreviated name of the amino acid that they code for. So let's say that the codon is GUC. So we would find GUC right here, and that's going to code for the amino acid Val. And it takes thousands of amino acids stuck together to make the protein, but that gives us a starting place. So how do you find what it's going to code for? How do you use this chart? Initially, you're going to start with your first letter over here, your second letter here, and your third letter here. So this time, let's say it's AUG. our first letter we're going to find on this side, on the left-hand side. So I find the A row. And then I say it's going to be U, so AU. So I'm going to find the column at the top where it says second letter that has U. And where these two come together, I should find AUG. And I do. And in this case, it stands for MET. And I want you to notice that the MET is in green. It's in a different color. And that's because MET, literally that amino acid, all it means is start. Start paying attention here. And then, of course, we have 3, UGA, UAA, and UAG. And these all mean stop. This means we've reached the end of the gene. Stop adding amino acids. Stop paying attention. Anyway, I'll see you next time on The Sound of Science.